animals. So as I was inclined to visit the water all the time, I also was exposed to some of these animals. And that was really at a young age, where a lot of my love towards aquatic animals appeared and developed through time. Nothing of this could have happened without the guidance and love first of these two folks in here. They're actually visiting here in California, so I have the great pleasure to have them, and I hope I shared the link with them, so I hope that they are hearing me. So my mother and my father both introduced me not only to the beautiful natures of Costa Rica, but also to science. Both of them are in the medical sector, so they deal with human diseases, but I was exposed again, since a young age, to science. And then, of course, as I grew older, I owe a lot of the support, and absolutely the tremendous, backup, that I have, at home, to my awesome wife and my three children that are the motive of my inspiration nowadays. So my story in science, really began, after high school. Again, I was inclined, into medicine, but one thing I knew was that I didn't want to deal with people or deal with people at minimal. So I didn't want it to become, a human doctor, like my parents, but actually my love, towards animals, started growing, and I learned about, research, the opportunity of research. So really was inclined about research at that point, So I was admitted and into the Escuela de Medicina Veterinaria in Costa Rica, where I had tremendous mentorship, particularly from my pathology professor, who, really was the first one, to open the doors, towards a career in aquatic medicine. In Costa Rica, you have to do a research project, before you graduate from vet school. And at that point, I try to merge, my love for the water and animals and research, and was able to start working with sustainable aquaculture, and particularly with infectious diseases, that were affecting at that point aquaculture in Costa Rica. So these are different settings of aquaculture in Costa Rica. Freshwater aquaculture, in some of them, more static systems, some of them in net pens, and some of them in flow-through systems. But at the time, there was a big outbreak of disease in one of these systems, particularly in this one in Tuetal, in Alajuela. This is young me, working, when I was in my senior year in veterinary school in one of those samples, So there was a big disease, that occurred that was the first time really that hit Costa Rica. So we had the opportunity to invite some researchers, from the US. And in the US, aquaculture, one of the biggest places, where you can actually learn about aquaculture is in the southeastern US. Specifically at Mississippi. And Mississippi counts, with one of the largest industries, of raised fish, specifically with catfish. So I had the privilege, to join the Mississippi State University College of Veterinary Medicine right after graduation. So I finished vet school in 2005 and joined immediately the CDM at Mississippi State, where I focused my master's in science research, investigating one bacterial pathogen that affects, freshwater fish, Flebobacterium columnari. So it was really at this stage, that I learned of my second love, let's say like that, for science. So the first one being aquatic animals, the second one actually being the microbes that infect, these animals. And I really started learning more on microbiology and research in microbiological sciences. So at the Tad Cochran National Warm Water Aquaculture Center, that is basically located in the mid of Mississippi, we had, I had the chance, to really learn about aquaculture and started working with a bacterial pathogen, that I still work on, almost 20 years after I started here. After my master's, again, and wanting to learn a little bit more about that one pathogen that was affecting Costa Rica, At the time, I had the chance, to join the School of Veterinary Medicine at Louisiana State University, where I had a little bit of broader scope, in terms of pathogen diversity. I continue learning about microbiology, but now I joined the pathobiological science department as a grad student, now pursuing my PhD, where with Dr. John Hawk and Dr. Ron Thun, I investigated, that one pathogen that was killing tilapia in Costa Rica. So we investigated a lot in more depth, Francisella orientalis, and not only identify the pathogen, but started developing practices, to prevent, these infections in vaccines. Basically, started looking at vaccines. So immediately after I received my PhD, I joined Ross University School of Veterinary Medicine. So this was actually my first faculty position. This veterinary school is located in the beautiful island of St. Kitts, which is in the Caribbean Sea. So I moved from the southeastern United States, to this tiny island, where although it is fairly tiny, it is, as other places in Latin America, extremely rich in biodiversity. So in here, I had the opportunity to not only expand on my microbiological interest, again, infectious disease that affect animals. We started studying, African green monkeys. We started, looking at some zoonotic diseases, that affecting, mongoose, rats, but also continue exploring opportunities, now with sea turtles, as well as a wide range of reef fish. So it was, during these 5 years that I spent at Ross, that I really had the privilege, to start interacting, a lot more contact, with veterinary students, and particularly with veterinary students, from a very, very diverse background. So in 2015, Destiny brought me to beautiful California, where I had, the privilege, to join the UC Davis School of Veterinary Medicine, where I currently work as a professor of aquatic animal health. I do the work, I have the privilege, to work there, but only because I have, again, the absolute privilege to work with a wide range of colleagues, from staff, to faculty, to the students. And the students, really are the ones that deserve all the credit, for the presentation, that I'm going to give in the next, half hour or so. But even with all of this capacity that we have in California, we really will be nothing, if it wasn't, from those strong interactions, that we have with other colleagues, and collaborators from other universities and institutions. And I put in here, the logos of just some of the excellent collaborators that our laboratory counts with, particularly because of the topic that I'm going to present today. So I have to recognize, for sure, the strong collaboration that we have with the California Department of Fish and Wildlife, with Dr. Mark Atkinson laboratory, and the absolutely great colleagues, that work, in this institution, that again are providing a lot of the samples, that we work with, as well as, other universities, Mississippi State University, Michigan State University, Oregon State University, USDA, IRS, and then of course, private industry that you will see where a lot of these collaboration, not only with state officials, but federal agencies, and also with other institutions, becomes critical when dealing with infectious disease. So what do we do in our lab? So our lab is fairly diverse, not only because of the race, ethnicities, that we have, but also on what we do. So we focus a lot on aquaculture, not only for fish that are destined for human consumption, just like tilapia or white sturgeon, but also ornamental, so like koi and goldfish. Additionally, we do a lot of work with conservation medicine. So again, working closely with the California Department of Fish and Wildlife, we can, develop a program, where we can serve also wild species of fish. We also have strong collaborations, with different aquariums around the area, with the Steinhardt Aquarium, for example, and Monterey Bay Aquarium, where we also have the opportunity to partner with them in teaching programs, so not only in research and clinical work, but also education. So we have a fellowship, that is completely focused on aquatic animals. And it is one of the unique ones, that are present, not only in the US, but worldwide. So we have also, again, a great opportunity to train veterinary students and undergrads and grad students here at Davis, at different capacities, so all of these, have been different efforts, from surgery, by residents and aquatic animal health fellows, to training of veterinary students, at different stages in their curriculum. We also serve the community by providing clinical service to pet fish and culture fish. So through the veterinary teaching hospital here at Davis, we actually can see fish, and we have treated them at a number of different conditions. And of course, research. So as I mentioned before, I have a particular love towards infectious disease, where we incorporate, not only, again, important pathogens, that can affect culture fish, but also those that affect wild fish species. And within this, I want to start presenting a little bit of what we do and how we approach, the different cases, that we might see. And when we work with fish medicine, we cannot escape, from understanding also the environment and the pathogen, of course, that sometimes, cause the disease in the host, So our lab focuses on diagnosis, treatment, and prevention of these infectious diseases. But to actually achieve these, we have to understand this interaction that the host and the pathogen have, so the fish and, for example, bacteria, viruses, or fungus, might have, and how that disease actually occur. So again, we study the bug or the microbe, but we also study, environmental conditions that allow these particular microorganisms to cause disease in these different hosts. So it is this interaction between these different elements, that actually can result in disease. But importantly, for us to understand how to prevent those diseases, we will have to first understand how is it that disease actually occur. And this is important, again, in many aspects, from conservation, but also with the growing human population, we have to recognize, that fish and fisheries are still basically a great amount of high quality of protein, that, again, this growing human population, requires. So if we look at this graph, from the FAO, you can see that how aquaculture has grown, in the last century or so. So you can see that, again, 50 years ago, 70 years ago, most of what was consumed by humans, came from wild captures. Well, we're becoming very, very effective fishermen and fisherwomen, and we have decimated many populations of wild stocks. With that, sustainable aquaculture has actually come, to provide tremendous aid. And nowadays, pretty much 50% of what it is consumed, by the growing human population comes from aquaculture. And again, that provides some relief, to these wild captures. So now focusing a little bit more into, again, the research project that I wanted to share with you. This is the brief outline of what I'm going to present. I'm going to introduce Piscine Lactococcosis, so that disease and how it actually came to diagnose, became diagnosed here in California. Then I'm going to present a little bit of molecular diagnosis and genotyping, that has been spearheaded, by different members in the laboratory. Present a small epidemiological analysis, that one of our excellent grad students performed a couple of years ago, and present some data on laboratory control challenges, virulence, and experimental vaccines, that we have developed. So this story of Piscine Lactococcosis in California is pretty new. So again, 2019 seems, so far away, but if we can, bring back our memories to what was happening in 2019, 2020, we cannot escape, the pandemic, right? That we still have around and COVID. So really anything else that happened during that time, was really on the background, from COVID. And of course, that is absolutely warranted. However, when it rains, it pours. And back in the spring of 2020, we started receiving, samples from the California Department of Fish and Wildlife, because they were concerned of abnormal mortalities that started appearing in 3 different fish hatcheries and 1 private aquaculture farm in Southern California. So these happened around Victorville, California, and there were different announcements, that had to made, because again, this was fairly unusual mortalities, and they weren't able to seed the lakes and the rivers as they typically do with the fish, that were present in this hatchery. So we, again, worked very closely with them, and they submitted some samples, to our lab for confirmation and research support. This is a map of California, basically showing all of the different hatcheries that the state has. And these are specifically for state hatcheries. Again, this is not even taking into account the federal hatcheries, but this is the area, and initially the 3 different hatcheries, that were affected, okay? So this is Ontario, California, this is Fresno. So in the East Sierras, and again, in Southern California, in Mojave River Hatchery. And these were the presentations, that again, colleagues from CDFW presented. So with fish medicine, again, you start looking at the external lesions, that the fish may have, so there will be what is commonly referred to as pop eye or an exophthalmia. So almost like all of the eye is bulged out of the orbit with redness, sometimes, inside of the eye. Some of these petechial hemorrhages, were observed, in the buccal cavity. The gills, sometimes, were pale, sometimes, you can even have some of those hemorrhages, within the gills. And there were changes in the color of some of the affected animals, so some melanosis, they will be darkened in color than your typical rainbow trout. Internally, all of these is fairly abnormal. So all of these are very, very congested vessels, which again, really points out, to some septicemia. So basically a massive bacterial dissemination within the bodies of these animals. At that point, we developed differentials that are basically just out of the clinical science. What is it that we might think, might be happening? Streptococcus, Enterococcus, and Vagococcus are some bacterial pathogens, that we have found in California in previous years. Of course, those become part of our differentials. But when we started looking a little bit closer into smears of these affected animals, we start finding this again, coccus sometimes, in chains. And one of the diagnoses, one of the samples that came up positive for this bug, Lactococcus garvieae, which Lactococcus garvieae is one that has been previously found. It is an important organism that has been reported in the Mediterranean, particularly in Europe and in some Mediterranean countries, but not really in North America. So at this point, we were pretty baffled as to if this was real or not. Come up, the summer of 2020, unfortunately, mortality had reached already 75% in some of the affected system. And because of these novel diagnoses, and because this organism has the potential to infect humans, and particularly because there was no control was identified at the time in terms of medicated feeding of these fish, over 3 million animals had to be euthanized, okay? So that was a big decision made by the CDFW, again, guided, by, again, what we knew at the time, from the outbreaks and the research. And of course, these, even during the outbreak, make local as well as national news, So you really don't want to appear in the New York Times, for the wrong reasons, but again, these actually made, important national news, because it was a big hit for sure for the stocks of fish, in California. So let's talk a little bit of Piscine Lactococcosis before we move too much into the research. It is caused by Lactococcus, and historically, as I mentioned before, it has been attributed particularly for this bacterial species, Lactococcus garvieae. These are gram-positive, chain-forming, non-hemolytic organism in blood agar. So pretty much, this is what the colonies in agar will look like. And this is what the bacteria specifically, again, if you look at it microscopically, would look like. It has a global distribution, again, some of these Lactococcus, and it has been previously found in other parts of the world, again, in culture and wild species, warm, temperate, cold water, fresh and marine systems. So pretty widespread bacterium. But again, we have to remember that these are opportunistically zoonotic, and there has been even review papers, associating the zoonotic potential of Lactococcus garvieae, and again, diseases in humans. In fish, the clinical signs and gross pathology that has been reported for Lactococcosis are pretty much, exactly the same as we saw in our case. So lethargic, exophthalmic fish, darkening of the skin or melanosis, and hemorrhages pretty much in every tissue in the body. And unfortunately, as I mentioned, even for other outbreaks, it has been known that this is difficult, to treat. So there is growing antimicrobial resistance, and there are references, to vaccine escape. So even when vaccines have been attempted, it hasn't really been that good. In the Americas, this is again a little bit of a timeline of what has been reported for Lactococcosis. So in Brazil, there has been reports, since 2007 of Lactococcosis, particularly in pintado, as well as in tilapia. But then in the past 10 years, is really where we see an increase of cases, first occurring, a single report in Washington state, but then big outbreaks occurring actually in 5 different farms in 2 different states in Mexico in 2016 to 2018. And then this outbreak that started occurring in 2020 in Southern California and the East Sierras. And since then, in the last 3 years, there have been always associated with the spring and the summer, big outbreaks occurring now in Arizona, as well as in Canada, specifically in Ontario. And talking with colleagues, from Brazil, in the past 5 years, even thought, the first report was made, in 2007. In the past, 7 years, there has been a significant increase of cases in Nile tilapia and pintado in Brazil. So one of the first questions, that we had, was, well, if this bug is now appearing, in California, are they the same? And again, I didn't know. So we started, focusing some efforts, in the lab, to clarify this. If you use what has been already published and is available for the diagnosis of this bacterium, you will find, that there's some molecular assays. So these molecular assays target a specific region on the bacterial DNA. And when we compared, the 4 different affected farms, so these are basically isolates that were recovered from the 4 affected farms, you will see that this band indicates, that yes, all of them are being detected. So we call it PCR positive. At this point, yes, it is Lactococcus garvieae, end of story. However, we have to recognize, that there is always some constraints, to just following what has been published, and again, assuming that all is known from, what it is published. If we look at biochemical assays, conventional sequencing of universal genes like the 16S, or even using, again, these available molecular methods, it indicated, that all of the isolates that we had recovered were pretty much the same. So Lactococcus garvieae. However, part of research, is exploring further. And I have to give credit, absolute credit in here to Dr. Taylor Heckman, who is a postdoc, at the Aquatic Animal Health Lab, in our laboratory, as well as Zainab Yazdi, who's one of the research associates, because Dr. Heckman was really the first one, to start, posing the question of like, is this the same or not? During her PhD here at Davis, Dr. Heckman was able to develop a specific molecular method that allows a good discrimination from a similar bacterium as Lactococcus in Streptococcus ine. And she uses, some different genes, than this 16S that is typically done. So what Dr. Heckman did was, in collaboration again, with Zainab, they first amplified the 16S. So this one gene from all of the isolates that we had recovered at that point. And all of them really indicated what this dendrogram indicates is that pretty much all of them are the same. However, if they use this discriminatory method, that Dr. Heckman developed, now we can actually see that targeting other genes, you see very, very, very obvious differences, So you start seeing this grouping in here, this grouping in here, and this grouping in here. So very, very different, than this homogeneous group that we will see in here. So they're in fact very different organisms. So these can be further explored by using whole genome analysis, where you actually sequence the entirety of this genomic DNA of the bacteria. So what Dr. Heckman was actually able to find was that what people were calling Lactococcus garvieae as a single species actually were 3 different species of bacteria. And this is very important, because if you start thinking about vaccine development, treatment, etc., it is important that it is targeted, to the right organism that you are trying to investigate. And this was fairly validated, by with collaboration with a USDA IRS, with Dr. Caitlin Alder and Dr. Geoff Waldweiser, as well as colleagues from Mississippi State University, Dr. Matt Griffin and Cindy Ware, where you can actually see that now if we look at isolates that were recovered from California, they share almost 90% or over 90% identity to Lactococcus petauri, whereas they only share about 92%, with, in fact, Lactococcus garvieae. So again, this actually gives us a strong evidence, to say that really what was causing the outbreak in California was Lactococcus petauri and not Lactococcus garvieae as it was first thought. So this is important also to recognize, because again, not everything that is in the literature is correct. And although this organism, Lactococcus garvieae, can still cause, disease in fish, in fact, most of what it is reported, even in the literature, as Lactococcus garvieae from fish are in fact Lactococcus petauri. So even within what we're seeing in California, Dr. Heckman has now acquired, over hundreds of isolates, so each one of these, not to bore you to death, with this very, very busy graph, but what I was thought, that's pretty cool was actually that you can see the source of these isolates. And again, these little rectangles or triangles or stars just indicate, what was the source, So these are the different hatcheries that now we know in the past, 3 years have been affected, with outbreaks of Lactococcus. Again, not only garvieae, but petauri and formosensis. And particularly, again, if you see all of these gray in here, all of that is Lactococcus petauri. And most of what you see in here in blue are the isolates from California. So now we know actually that the most important of the agents causing these very, very emergent outbreaks in California are in fact Lactococcus petauri. So at the time, where we started, describing, Lactococcus, as the causative agent of disease, there were some folks, that will come in and say like, well, but are you sure, Lactococcus is pretty much everywhere, even in yogurt. And, they're not that virulent. This is probably just another hoax, of science, fake news, everything is fake news, right? Well, are they? So we started, working again, with challenge models. Dr. Eric Littman, who is a previous aquatic animal health fellow. So he's a veterinarian, that participated in this fellowship, through the lab. Started testing Lactococcus petauri in the top, and then a Lactococcus garvieae in the bottom. And we infected fish with 3 different doses of the bacteria. So 10 to the 1, 10 to the 4, 10 to the 7. Again, what you have to know is that this is a lot of bacteria, the 10 to the 7, whereas 10 to the 1 is only a couple hundred, bacteria. Okay, we injected fish with these doses, isolates, again, the Lactococcus garvieae and the Lactococcus petauri, and we monitor the health of the fish, for 15 days, culture at 18 degrees Celsius. And what you see in here, this graph presents a percent survival. So again, if it wasn't pathogenic at all, there will be a 100% survival. And what you see in here is that these Lactococcus petauri that we were recovering from rainbow trout in California, all 3 doses caused close to 100% mortalities in only less than 14 days. So it is a pretty virulent strain, this L. petauri. To further understand, again, if the conditions, the environmental conditions, as I mentioned to you, these bugs started appearing in the spring. So what happens in the spring? Well, the weather starts changing, from the cold winter into the warm summers. And again, if you have spent, more than a couple of years, here in California, you will already have noted that the summers are getting longer and longer and warmer. So this is an effect, not only in the human population, but of course in aquatic animals like our native salmonids. So what happened in here? In this experiment, Dr. Littman inoculated this bacterium, the Lactococcus petauri that was recovered in California, inoculated trout that had been raised at 13 degrees Celsius or at 18 degrees Celsius. So there's a difference of only 5 degrees Celsius. And again, with the same amount of bacterium, and monitor their health, for 15 days. And what you can see in this graph is that hot is very bad, cold is actually okay. So what this means is that if you notice in this graph, regardless of the dose, 10 to the 2nd, 10 to the 4th, 10 to the 6th, if the fish were at 13 degrees Celsius, that is a temperature that they're supposed to be, there will be actually little mortality, close to non-mortality. However, as this temperature raises, as we're seeing it now with climate change, very, very high mortalities should be expected, from 70 all the way to 100% mortality. And the ones that don't die from the bacterial infection actually will have a pretty high persistence. So the bacteria itself will still be present in the survivors. And these can become shedders that can then, infect other fish. So if you think about it, how lethal this organism is to trout, around 20 colonies. And again, if you remember, the photo that I show you of how these guys, look in a plate, there will be, thousands and thousands of colonies, basically in an infected animal. So there were a lot of unknowns. What is the distribution of this pathogen? Where did it came from? How virulent it is, to other species? Is it everywhere? What species of wild fish, may be affected? Where can we find it in California? So a lot of unknowns, and again, we have again a pleasure of working also with bright young minds. Taylor Abraham is a master of preventative veterinary medicine. So she's currently a veterinary student, a third-year veterinary student, that took a break from vet school to focus on research. And she's working on her lab, to try to answer some of these questions. So before we get in depth, with Taylor's work, initially, Dr. Shaheen, another postdoc, in the lab, and SADAP, developed and validated a method, a molecular method that allowed the detection of DNA from all of these 3 different agents, that we know now can cause Piscine Lactococcosis. So they developed this method, particularly to be able to not only provide a rapid diagnosis from the bacteria itself, bacterial colonies, but also from fish tissues, and very interesting, from environmental samples. So with this assay, you can collect water samples, soil, even fecal material from birds, which we were suspecting were some of the vectors of this bug, and extract the DNA and then test for the presence of the bacterial DNA in these samples. So it provides an opportunity for surveillance and as well as diagnosis. So what Taylor did was she wanted to conduct a surveillance study to test for the presence of these agents that can cause Piscine Lactococcosis in water, so lakes, so that surround the hatchery that was first affected by this bug back in 2020. Additionally, she wanted to compare the virulence, so all of the isolates of Lactococcus garvieae, Lactococcus formosensis, and petauri that have been recovered from fish, wanted to better understand the virulence of these isolates into relevant fish species that we're going to talk in just a second. So what we did, so in September 2020 we basically partnered with CDFW and collected about 360 wild fish from these 4 different lakes that surrounded one of the affected hatcheries in California. So again, this is the map of California, and this is our sampling site. So this is Victorsville, and these are the 4 lakes that we sampled: the Jess Ranch Lake, Victor Valley College Lake, Spring Valley Lake, and the Horseshoe Lake. And again, this is the Mojave River hatchery that was one of those hatcheries that had to be depopulated due to lactococcosis in 2020. So fish were collected by CDFW and submitted to our lab for analysis. So we received a lot of fish, and it was then time, to sample them. So we collected samples from the kidney and from the brain, and we inoculated agar media, so to allow for bacteria to grow, but also use that molecular method that Dr. Shaheen had developed that again will test for the presence of DNA of these bacteria. So again, just to present some of the highlights of this study. 2 different Lactococcus garvieae. So now these are Lactococcus garvieae, the real Lactococcus garvieae, were isolated from what we have to call healthy brains of 2 different largemouth bass. Lactococcus, so either one of the 3 species that can cause lactococcosis in fish, were recovered only from bluegills and largemouth bass that were sampled out of 3 of the 4 lakes. But all of the lakes, the environmental samples from all of the lakes, particularly the soil, tested positive for these potential etiologies, that can cause Piscine Lactococcosis. So what Taylor did next, was actually investigated what was the virulence of all of these different strains that we have recovered through the past, 3 years. So we had 17 strains of Lactococcus petauri, 2 strains of Lactococcus garvieae, and 4 strains of Lactococcus formosensis. Again, just a reminder, all of these were found from fish, okay? So all of these were isolated from fish at different points in different places. But because we had isolated Lactococcus garvieae from largemouth bass, we decided, to do the challenge not only in largemouth bass, but also in not only rainbow trout that we knew were susceptible to the bacterium, but also in largemouth bass. And again, monitor for 14 days and test the survivor's brain and posterior kidney to investigate the prevalence of infection in survivors. So what you see in this graph are the 17 strains of Lactococcus petauri, the Lactococcus formosensis, and the Lactococcus garvieae that were evaluated. So very, very important to note, these on top are the California outbreaks. So these are the ones that we are recovering from California. No isolates of any of these bacterial species were found, to cause any type of disease or death in largemouth bass. So it seems like largemouth bass are actually resistant to these bacteria. Some of them, only 2 persisted, around 10%, so very, very minimal persistence. However, look at the mortality, overall mortality that was found in rainbow trout, about 64%. So most of these isolates were able to cause over 90% mortality in rainbow trout. So really, really indicating the Lactococcus petauri, it is a pathogen of concerns, for rainbow trout. Okay. So we know now that largemouth bass, seems resistant, but rainbow trout seems very, very susceptible. So what about other important species that we have in California, and particularly that are cultured in California? So let's take a look at rainbow trout, again, as a control, if we can put it this way. Chinook salmon, which is, a keystone species in California, extremely important species, again, for the ecosystem, and a native salmonid of California. Nile tilapia, one of the most important worldwide culture cichlids. The white sturgeon, again, another native fish species, in California. Absolutely beautiful fish, and one that it is important locally for aquaculture in production of caviar. And then koi, which is, again, an example of a carp that it is culture and maintained in California, particularly for the ornamental trade. So what we did in here was, again, we know our Lactococcus petauri from California, pretty hot strain, very virulent for rainbow trout. So we compared them side to side, to these other fish, monitor for 14 days, and again, sample survivors. And what we can observe, from these challenge models, was that really the clinical sign and gross pathology only occurred in the rainbow trout. So this is what a normal, rainbow trout fingerling should look like. This is what an affected one will look like. So you can see the changing color, the melanosis, and then the pupille, the exophthalmia, sometimes bilateral, sometimes unilateral. Again, what a normal, coelom should look, from a rainbow trout. So this is the liver. In here, the pyloric cecum, you can have here some fat, the spleen and distal intestine. And these are different grades of affected fish, where you can see, again, these hemorrhages, sometimes, fairly diffuse. And again, pretty much, present in every one of the organs. So again, a true septicemia was observed. If we look at the percent mortality within these animals, then you can rapidly see that, yes, this is a very, very concerning disease, for rainbow trout, but it can also become, an important disease for Chinook salmon. So there was a significant difference in the mortality pattern between rainbow trout and Chinook salmon, but all of the other species, were pretty much, I don't want to say resistant, but significantly less susceptible to this bacterial infection, when compared to rainbow trout and Chinook salmon. So these at the end are the numbers that we can see. So 96%, 56% cumulative mortality in the 2 salmonids that were tested, but 0 in all of the other species. So is there anything that we can do? So I have been talking, now for close to 45 minutes, and it is a pretty, bad story, right? Nobody wants to hear about, everything negative. And this is what my family looked like, for about, 4 years that I was coming only with bad news, So even the dog, was having a hard time, with me. But then again, it's hard to find, good news, in the news, but at least in this article, they presented, some of the moving forward efforts. What can we do and how can we help the community? And this is in vaccines developments, So in aquaculture, again, similar as to within humans, as well as in other veterinary sectors, there is different ways that we can prevent disease, from using immunostimulants, prebiotics, probiotics, and then vaccines. Yes, we can vaccinate fish. These are the first group of animals, that develop an adaptive immune response. So we can give injectable vaccines, or we can give mucosal vaccines, typically either by immersion, like in this photo, where you immerse a group of fish, with a vaccine, or orally. So that's what the goal, with this research, is to later on, hopefully develop, just as again, and this insert, from Merck, a vaccination program, just as we do in humans and in animals, where we can vaccinate at different stages, again, important, disease that the animal might see at different stages. So our first objective, again, spearheaded by Dr. Lindman, was to evaluate the protection conferred to rainbow trout by two different vaccines that, partnering again, with the industry, we were able to obtain, so the industry can formulate autogenous vaccines that actually are now being used, very excitingly, to prevent this in different sectors. But we tested immersion vaccine and injectable forms. And then with the excellent work, from a visiting student from University of Pennsylvania in Teresa de Routier, she did a program, where they will come into my lab for the summer, perform a research project, and then go back to school in the fall. So Teresa then tested, if booster vaccination could actually enhance the protection afforded by some of these vaccines. So what Dr. Lindman did first, again, was immunize different groups of fish, either using the injectable vaccine or the immersion vaccine. And then after immunization took place, he challenged, these fish with the virulent strain of Lactococcus petauri and monitor mortality and morbidity throughout 15 days. So if we look at how the immersion vaccine alone did, you can see that there was some protection conferred by the whole killed vaccine, but it wasn't great. It gave a relative percent survival of 28%, where we want these relative percent survival to be 99, 100%, right? To be 100% protective, which is really not realistic. Anything, I would say above 80 is very, very, very good. But what was worrisome, was that, again, there was still also some persistence in vaccinated animals, but still it did outperform, just the adjuvant or the positive control, which was, non-vaccinated. So this immersion vaccine did provided some protection, although it wasn't great as the intracelomic vaccine did. So the intracelomic vaccine, did had a conferred significant protection to the vaccinated animals, resulting in a relative percent survival of close to 90%. So very, very effective vaccine, and almost, or I would say, no persistence, in the brain or the posterior kidney. So then Dr. Lindman and Teresa also investigated basically the immunological response, that was conferred by the vaccines. I will not bug you too much, into this because of interest of time, but the vaccine alone, provided, more, of course, immunostimulation than the adjuvant or, of course, the non-vaccinated animals. Then Teresa wanted to explore if boosting vaccination will actually help. So in her experiments, she immunized every, 4 different vaccine treatments with the immersion vaccine. And some of them received a boost by immersion. Some of them received a boost by injection. And then we have the positive and the negative controls. And again, what Teresa did was, again, if you see this diagram again, immersion vaccinates some treatments, and then some of them got an injectable vaccine, some of them got an immersion boost, and then challenge them by cohabitation. And cohabitation is another model that we have explored, in which you have more of a natural route of infection to the fish. So you infect some fish, and then these become shedders, and you basically monitor the shedders and the naive fish and how they respond, actually to a particular treatment. So in this experiment, what we found was that the shedders, all of them were effective in, again, getting sick and shedding the bacterium. So there were not difference, between treatment. However, really only those that were intracellularly injected with this vaccine, were really protected. So although initially the immersion vaccine does provide some protection, this protection, it is very, very short, and really for a strong immune response, to be developed, using this type of vaccine, a wholesale kill vaccine, the injection is really needed. So in conclusion, from these vaccine studies, again, both modalities, the immersion and the intracellular injection, were found, to be able to provide an immune response, in the fish. But really, it is that intracellular injection, the one that significantly produce a protective response, and is again, now what it is currently used, in CDFW and in the aquaculture facilities. So is there any hope for treatment? Again, now we have undergrad representation, in here. We have, very, very, very smart, Isabella, in here, Alex, just graduated. And then Kelsey also helping, Dr. Heckman investigating potential treatments. And for here, again, we're looking at different antibiotics that could potentially be used by the aquaculture industry. Again, we have a panel of Lactococcus, petauri, formosensis, and garvieae that we explored the susceptibility to different drugs. And it is interesting, because they have different profiles, So one important thing, to note is that unfortunately, it seems like these very, very virulent strains, that we have in California, they do have resistance to some of these antimicrobials, particularly oxytetracycline, that it is one of the 3 FDA-approved drugs, to use in aquaculture. So this really presents a challenge, moving forward in terms of treatment. However, there is, again, we got to see the good, right, with the bad, in that there is potential for fluorophenical and erythromycin. There are 2 other drugs, that could potentially be used in aquaculture. So that is now what it is, a strong focus of our research. And besides that, we're continuing to investigate other modalities of vaccines, particularly for that immersion and oral route. So we're developing live attenuated vaccines. We are exploring, again, how is it that these bacteria persist in the environment, and the susceptibility to different disinfectants and antimicrobials that could potentially be used either to cleaning completely the facilities or as potential treatment for outbreaks. All right. So with that, I will close. I don't know if we have time, for some questions. I want to thank, again, for sure, 2 of the bigger sponsors, for, funding agency for our laboratory, and a close, with a big thank you and a pura vida for everyone for sticking around and listening to me. Thank you.
That was a very interesting talk. So first, I want to see if among the participants are joining us by Zoom. If any of you have a question, you can put it on chat so I can ask Dr. Esteban on your behalf.
Yeah, in the meantime, I have a question. So I know you mentioned that Lactococcus is zoonotic. I was curious what the transmission pathway might be for humans, whether it's like blood to blood or through ingesting.
Excellent. So that is an absolutely great question. If folks, in Zoom didn't heard, the question is, what could be the potential transmission routes, from the fish, into humans? That really hasn't been explored. There hasn't really been either a mammalian model. What has been suspected, by the type of disease, that has been reported is that oral route, has to be has the potential, and then iatrogenic, so basically puncture, wounds, from potentially fish or fish hooks, they can actually induce cellulitis or an infection. And what it has been, again, a little bit difficult to assess, is really how virulent will this bacteria will be in humans, because again, most of what has been reported, for mortalities has been already immunosuppressed hosts. So either, very elderly or folks that have some other conditions, that might make them more susceptible to really any bacterial infection. But there is of course, potential also to start investigating this forward, using some of the methods that we have presented in here for fish. We could explore that as well, using potentially a mammalian model, a rabbit or a mice, etc.
Thanks.
Yeah.
Yeah, I actually have a question related to climate change and also the idea of how to integrate, as an example, machine learning, like we're looking for algorithms that eventually could prevent, like, or at least to give a window of opportunity to prevent spreading of these zoonotic bacteria when you see that, okay, there is these weather conditions that we know that are going to happen or are changing more dramatically or faster than they did probably a decade ago. If with the help of artificial intelligence or machine learning, we can somehow, tap on this so we can know, okay, this is happening. So that means that in 1 week, we need to go to the fishery and do this assessment before it get out of control.
Yeah, no, that's an excellent question, and really in an area where aquaculture as a whole, has grown. The U.S. It is not one of the leading aquaculture countries, but for example, in Norway, in Canada, and even in Chile, there has been a lot of efforts, to again, have developed, epidemiological models, for example, for some of the diseases, that are, really known, to be present. In this case, we just knew about this, not even 3 years ago. So there has been a lot, that needs still to be learned, but through some of these some of these data that has been acquired, particularly with better understanding of the conditions, hopefully, that allow this bacterium to cause the disease, like the temperature, as it was mentioned, how long, can it persist, in the host or even in the water and in the soil. I think all of this information, could be utilized, later on, by epidemiologists, to then generate, the models. There has been, some research done in other organisms where, for example, they use molecular methods, to investigate, the load of the pathogens, in different environments. So there's one, for example, for a parasite that affects salmonids, where they know that there are hot pockets, throughout the rivers where, for example, due to the presence of dams, these fish, cannot move too much, and they might be present in these, like, hot pools of areas with a high load of the pathogen. So they use molecular methods, to basically quantify the amount of these organisms throughout time. And they know that there is a threshold. When that threshold is made, that they know that if they keep the fish at those conditions with that load of the parasite, there will be an outbreak. Well, then they actually have, buy-in, from officials, state and tribal officials, to open the waters of those dams. And then that basically dilutes, the amount of the pathogen and provides better conditions for the fish. So there is data generated, in that sense, from other organisms. We are not there yet for Lactococcus, but it is clear that, climate change, does play a role with this disease, particularly, again, because of the time of the year that we see it. Again, and coping that, adding that, with the data that has been obtained, from laboratory control challenges, where clearly the temperature, again, is it affecting the fish, or is it, basically inducing something in the bacterium that allow for that disease to occur? That's still up to the debate, and more research is needed to figure that out. But yeah, that the role of the temperature really is is well characterized now.
And if there is no question in chat, I would like to ask another question. It would be in terms of wildlife, not so much in terms of other fish, but knowing that there are so many birds of prey that, go for fish. I have been many times at the Nimbus hatchery, and I can see the osprey and the eagles and the owls trying to get some fish out. Do you have any data about how these particular pathogens could also cross into the avian species that fish for their food?
Absolutely, that's another great point. Actually, our initial suspicion, was that piscivorous birds are very likely what brought this bacterium in the first place to this area with the migratory patterns. Again, where our initial suspicion was that probably due to the outbreaks either in Washington state or in Mexico. Again, we're right in the middle from migratory birds routes. So it is very, very plausible, to think, that it was that. Now we know that still, like, that doesn't really explain the whole, that doesn't bring, like, the full explanation, because we know that our isolates are thought they're very, very closely genetically to the ones in Mexico. They're different. So they provide a different antimicrobial profile. So they're slightly different to the ones in Mexico, a lot more different to the ones in Washington. So we still don't know the role that piscivorous bird might have, but we do know we can detect the bacterial DNA from birds' fecal droppings. So if you noted in one of the papers, that was published, by Dr. Shaheen, when they developed, the qPCR, when we developed the qPCR, one of the first, materials that we wanted to test, were the fecal droppings, from the birds, and that actually allows us to start, presenting the hypothesis of the role of those birds. Still, it is not, again, it hasn't been investigated with an avian model also what could happen, to the bird. But I can tell you that, again, the Lactococcus, what has been thought to be Lactococcus garvieae, again, very likely other species. There are isolates that have been recovered from avian species. They have an isolate that have been recovered from humans and other animals. So it is not only that transmission between, fish and humans, but there could also be, cross transmission, from, to avian species, other, again, piscivorous mammals, and whatnot. So yes, it is a disease of concern, not only for fish, but what could it cause, to other animals? It's still unknown.
Thank you.
All right. Well, thank you very much, everyone. If there's no more questions, I appreciate you guys attending, and I wish you a happy afternoon.
We have one question. We have a hand up from Amelie Segata. You want to call on her? Or?
Yeah. So do you have a
Amelie, feel free to unmute yourself and ask your question, or put it in the chat if you'd like.
Yeah, I don't see the questions in here. So
okay, let's see. She says, do you let me unmute myself. She said, do you have data about a potential vertical transmission?
Excellent. So again, that's another excellent question. We don't. Let me start, by saying that there are some bacterium, and some viruses, that are known to be vertically transmitted. So vertical transmission for folks, that don't know, is basically transmission that could occur, from the broodstock, so from the mother to the eggs, or to the fry fish. Okay. So there again, there's premises, for that, that has occurred, with other pathogens, not really for Lactococcus, unknown. I don't think, that it is crazy to think, just because of the septicemia that we see it happen in affected fish. We pretty much, can isolate it, from any tissue that we sample. However, the reason why I do not know if this is going to be, biologically relevant is the virulence, So at least in the rainbow trout, most of the affected animals at those conditions, will unfortunately die. And those that survive, if they develop a protective immunity, they are typically, they can shed the bacterium, but not really get infected. And that loads of bacteria will go to some specific tissues, like the secondary lymphoid organs, like the spleen, the spleen, the kidney. And very interestingly, we suspect, that it goes, also to the brain. How much of that bacteria can go to the gonad, where again, one will suspect, that will have the loads, needed, for vertical transmission to occur is unknown. I don't know. That will be an interesting experiment, to conduct. But I think, still that at that point, really the shedding and the horizontal transmission, will be more biological significant.
Yeah. So I have just not a question, but a little bit just to spur your what is your interpretation or your idea about, since this campus about, diverse perspective in sciences, what do you want to talk about or spur a little bit to inspire the audience about, in one side, have an open ear to the native population or indigenous populations in the US that have a very particular relationship with nature, and how we can learn from them about how to take better care about, let's say, our fisheries and etc. But also the idea of globalization, like, California, you have a beautiful slide showing all the different institutions in the US and out of the US that have been collaborating in your efforts, also the idea of, hearing what the native and indigenous populations here in the US but elsewhere have to say about this, but also, what other communities could impact your research.
Yeah, and that's a great point. I wish, I could say that there is a strong, work, everywhere. That is not true. Coming from Latin America, there is a very, very, very few basically crosstalk, between I would say, state officials, and native populations, that might be affected, by some of these diseases. Here in the US, I would say, that it is a little bit different, or at least, there is some glimpse of hope, and the reason why is that actually in the Pacific Northwest, tribal communities actually do have veterinarians, that can be hired, by them, to oversee, for example, some of their hatchery programs, some of their conservation efforts, so there is, some crosstalk, at that point. And there are organizations, for example, the American Association of Fish Veterinarians, World Aquatic Veterinary Medical Association, that actually have these folks, being members. And it is absolutely great, because then there is at least, some crosstalk, at the level of science, diseases, as they appear in some of these species, that of course, can have a big impact, into native populations. Can I say that happens, in Latin America? Not really. In terms of globalization, yes, absolutely. It is nowadays, extremely easy to get, animals, samples, frozen, or even live animals, from different sectors moved, throughout countries. Different countries have, different legislation. Different states even have, different legislations in the movement of live animals and subproducts. So, the best I can say, is to go by science. What is known, through science. There's been, again, of course, there's always push, from different sectors, to like, oh no, this is too regulated. Other ones say like, oh no, there's so much regulation that this is not conducive to business, but there has to be a middle point, and crosstalk, from those sectors, really to prevent, dissemination of disease. And that is hard, that is hard, because, really being a regulatory body, I wouldn't want to be in their seats, for sure. I work with them, but I work with both sides, and it's a complicated discussion.
Okay. Yeah, so thank you so much, and thanks for the audience. I don't see any other question on the chat, so well, I see a chat. Oh, okay, no, yeah, someone just thank you for the interesting talk.
All right, excellent.
Mhm.
Thank you for the invite, and again, looking forward for future talks.
Thank you.
Bye.